to do to make that a fruitful meeting. So now I'd like to introduce Erin. We've already had a little introduction there, but she's the genetics technician for the apiculture program at NC State with Dr. Tarpey. And she's also the primary for the disease arm of the Queen and Disease Clinic, as well as working for other lab and field projects for the program. And I think it's going to be this same program at the last NCSBA conference. And I'm telling you, it was the highlight of the conference for me. So very impressive. So we're going to hand it over to Erin to let her do her thing. Thank you.
Well, thank you so much. I really appreciate that. I'm going to start this presentation. You guys can see that just that PowerPoint slide, not anything else, right? Good. Awesome. All right. So as was mentioned, I work at NC State with Dr. Tarpey. I've been with him for 5 years now, working in that program. It's my dream job. I love it. And so what I'm going to talk about with you guys today is a specific project that I've been working on for the past couple of years that has really sort of become my baby, and I'm very excited about it. And if any of you were at that last NCSBA meeting before we were no longer allowed to meet in person, you will have seen this before. So I can see the chat, but I can't type in it. I did start recording, so I have the answer to that question. All right, so just a little bit of what we're going to go over today. We're going to start with an intro to Varroa mites, which will be old news to a lot of people, but it's really interesting to me that sometimes even seasoned beekeepers haven't seen like the life cycle and things like that. So we're going to do a little bit of review. Then I'm going to talk about honeybee viruses, which the longer I've been working with them, the more familiar I find beekeepers are. But we're going to go over some of the ones that you guys hear a lot and what they look like and what to look for and things like that. And then I'm going to shift the focus into this paradigm that we've created, this sort of overview of the research that we do and different projects that fit under it. And then we're going to really narrow it down into this specific experiment that I'm discussing today and what came out of that and where we hope to go with it in the future. So to start, there we go. Varroa is a parasitic mite of honeybees. It feeds on the fat bodies. That's relatively new research that has come out. Previously, this slide would have said it feeds on hemolymph, but we've found more recently that it feeds on the fat bodies. The actual mite reproduces in the honeybee larval cells. So those phoretic mites that you see on your adult bees, that's not where they typically reproduce. They reproduce inside capped cells. They can spread disease and weaken colonies, which everyone here I know already knows. Something I always like to mention is that often when you're seeing mites on adult bees, crawling on a frame and things like that, it's a sign that your mite levels are very high. You typically don't want to wait for that visual signal before you start investigating your mite levels. And we control Varroa chemically and by intensive beekeeping. I try to be pretty careful to talk about Varroa control and not specifically Varroa treatment. I know that there's a lot of trains of thoughts on Varroa control, and there's a lot of different ways to do it successfully. And so really what's of most interest to me and my perspective here is that you are controlling for Varroa in some way, shape, or form, whatever that looks like in your operation. So I touched on this a little bit already, but for anybody who's not familiar, the Varroa life cycle starts when a queen lays an egg in a cell. That egg will hatch into a honeybee larva, and then that mite who's ready to reproduce will enter that cell and actually hide beneath it to hide from those worker bees. And then once the cell is capped, the larva has grown big enough, the cell is capped, that mite will feed and reproduce, and those new baby mites will also feed. Ultimately, assuming that it survives, that bee will pupate, mature, and eventually emerge. Or alternatively, it will not successfully make it to adulthood and it will be removed by workers or by hygienic behavior. And when that happens, whenever that uncapping happens for whatever reason, that's when those adult mites will leave the cell. Anytime the mites are feeding, viruses can be transmitted. That's really the takeaway of that whole cycle. So whether it's on the young or on the adult bees, if mites are feeding, viruses can be transmitted. So I already touched a little bit on mite control. I really, really like this graphic out of Penn State, which they call the IPM pyramid. IPM stands for Integrated Pest Management. Anybody that does any kind of growing or farm work might have heard that term before. And so this really has this sort of this scope from prevention up to intervention. And then towards the top of that, we've got an increasing scale of toxicity. So with prevention is when you start with your resistant stock, your small cell comb, your manual brood breaks, things like that, which is sort of, it's not a direct Varroa control necessarily, it's just trying to keep those levels low in the first place. And then above that, we've got our mechanical control, which is going to be your mite trapping, screen bottom boards with sticky boards, powdered sugar dusting, if that's something that you do. And then beyond that, we're going to start getting into chemical methods. So sort of the less toxic natural compounds are going to be your formic acid, your oxalic acid, thymol, which that's usually your Apogard, things like that. And then sort of as that worst case scenario, last ditch effort, you typically see those synthetic compounds like the amitraz, which is sold as Apovar. And so what a lot of beekeepers experience if they do use chemical control is they move their way up this pyramid as the mite levels increase or as their difficulty with mite problems increase and things like that. So switching gears a little bit, what is a virus? This is actually really funny. This is another slide that before COVID, I think was a lot newer news to a lot of people, but now everybody's been talking about viruses for months, and I think that a lot of this is review at this point. But viruses are biological agents that cannot reproduce without the help of cells of a living host. So you may or may not have heard arguments among really esoteric science types like myself about whether or not viruses are alive. And that's the reason, is because viruses need those cells of those living organisms in order to reproduce. When a virus infects a host cell, it hijacks the machinery of that cell to make copies of itself. It sort of takes over and it forces it to do its bidding instead of what that cell would typically do. Over time, viruses can change, mutate, and evolve. Again, I think that's maybe a little bit old news. That's how you get different strains and things like that. You get antibiotic, not antibiotic, but antiviral resistance and things like that. There are a lot of different kinds of viruses with different structures, transmission routes, and hosts, including plants and animals. And a lot of viruses are very, very specifically adapted. Some can, infect a lot of different kinds of, say, plants or a lot of different kinds of animals. And then on the other side of it, you've got a virus that only will infect one very specific organism. And that's the only place it can be successful. So really a lot of variety in those viruses. And I accidentally touched on this. Antibiotics don't kill viruses because that mechanism that kills a living organism doesn't work on them. Antivirals and vaccines are typically used to combat viruses, especially in humans, in human medicine. So virus transmission is something that's important to really understand as we go forward in this talk here. And there are two kinds of virus transmission. There are two routes. The first is vertical, which is very simple. It's just passing of a virus from mother to offspring, in this case, the queen mother to the offspring. The virus can be passed on the outside of the egg or within the egg, but ultimately the mother gives it to the progeny. The other mode would be horizontal transmission, which is between workers or drones or by mites in our very specific case. So that can happen by trophallaxis, where you see bees, feeding each other tongue to tongue. We've got food contamination. So if there's virus located in bee bread or in honey or anything like that, it can be passed that way. Caring for larvae, where you've got nurse bees who are attending to those larvae, virus can be passed that way. That would be horizontal transmission. Mating of drones with queens, there's some really interesting work that Dr. Eshmail Amiri does out of UNCG about the passing of deformed wing virus during mating. It can be airborne. I am not aware currently of any honeybee viruses specifically that are airborne, but the example I fall back on now is COVID. And then feeding by mites, which is what we're going to talk about today. So that's a form of horizontal transmission of viruses. So a little bit more about honeybee viruses. There are more than 30 viral pathogens known to infect honeybees, and as science progresses and more investigation is done, especially genetically, we find new ones all the time. Viruses are detectable in the lab in adults, brood, mites, and even in the brood food and comb. So there's a lot of different places virus can be sequestered. We're still learning about their effects and interactions as a science community, and that's ongoing work. And so a lot of the times when people ask questions, really excellent questions, we don't know the answer yet because there's more work to be done to understand it. really specifically, we don't have definitive answers about what viral presence or viral level means for colony health or survival a lot of the time. We know that it's a stress on the colony. We know that there's an association with colony weakness and colony loss, especially with certain viruses. But unfortunately, we don't yet have the answer of, this amount of virus is when things start to become problematic, which is something I'm specifically pretty interested in as a research goal. There's no definitive treatment for honeybee viruses. The best thing that you can do to manage viruses is to manage your mites. And some of the viruses of interest that I'm going to talk about in a little more detail are sac brood, black queen cell, and what I'm going to call the paralysis viruses. So sac brood, a lot of beekeepers may have seen this before, even if you did maybe not know what you were looking at the time. This gets abbreviated as SBV. With sac brood, you're typically going to see perforated sealed brood, so like just an opening in that capping sometimes, or a total uncapping. There's going to be jagged edges because the bees chewed that open, and the larva inside is going to be canoe shaped. That's sort of one of the telltale signs. If it is a pupa, it's going to be present with a shrunken head. The larvae change in color with different levels of infection and over time, from the pearly healthy white to a pale yellow to brown and eventually to black, until finally the brood is brittle and easily removed. And this is most commonly mitigated by requeening. It's not considered a very serious infection on its own, but especially in a weak colony, it can spell death. Black queen cell virus is of most interest to anybody that does queen breeding, that rears any of their own queens, even on a small scale. This one gets abbreviated BQCV. In this, we're going to see those queen larvae turn pale yellow with tough skin until they eventually darken to brown and then to black. And a lot of the times, even if you don't open up that queen cell, you can see a dark spot on the outside, which is a telltale sign of black queen cell virus. A low take when you're grafting can also be a sign of black queen cell because it causes those queen larvae to not develop. Workers and drones can be infected with this virus, but they don't show any symptoms. And the reason that that's important is it means that the workers can pass this to potential queens, even though they don't appear sick, because they can hold on to that virus and pass it on. Deformed wing virus is sort of the big name right now. It's got the strongest association with Varroa mites and the strongest association with colony loss that we're currently aware of. There are three, I'll call them strains, that are currently known: deformed wing A, B, and C. The different strains differ in virulence and distribution. There's some really interesting work that suggests that with higher mite loads, deformed wing B starts to become more prevalent and deformed wing A becomes less prevalent, things like that. Those are relationships that we're still investigating. The symptoms of this are pretty straightforward. You see distorted, misshapen, twisted, wrinkled wings. Sometimes they'll even be balled all the way up. It almost looks like balled up tissue paper on the back of a really infected bee. Those visible symptoms are often absent or rare until a colony is highly infected, which I mentioned with the Varroa mites, that if you see them, things tend to already be pretty bad. That's often true with deformed wing virus as well. And as I mentioned, this has a very strong association with Varroa. They're very closely correlated. The paralysis viruses, there's a handful that I'm going to talk about today. The first get grouped together into what is called the AKI complex. That's acute bee paralysis virus, Kashmir bee virus, and Israeli acute paralysis virus. Then sort of in different categories are chronic bee paralysis virus and the Lake Sinai viruses. This is a video that I like a lot. One of the inspectors, Lewis Cobble, sent this to me along with a sample a couple years ago, and it did turn out to be CBPV specifically. But what I really like about this video is it's a really good example of what we tend to see with these paralysis viruses. So we're looking at this bee on the top of the frame, and we're seeing shaking, uncoordinated movements, shivering is something that gets described a lot. And something that's worth noting is that these paralysis viruses often look a lot like a pesticide kill. A lot of the symptomology is the same. So it's important when you do suspect a pesticide kill or a viral infection to call your local inspector and have it investigated, because a lot of the time it's impossible to know what you're really looking at until you get it tested. So the AKI complex, I already told you, it covers those three different viruses. These often don't display any visible symptoms. When you do see symptoms, it's going to look like that video, that shaking and shivering. With the paralysis viruses, you typically will see dead bees below the entrance. That's because they don't survive very long if they're highly infected, and so they die, and they're either carried out of the colony by undertakers, or they will remove themselves from the colony right before they die. These are strongly associated with colony death. They're highly virulent. When they take foot in a colony, it can get really bad really quickly. And these are transmitted by Varroa as well as other routes. So it may not have that same very strong association that deformed wing does, but they're absolutely transmitted by Varroa. Chronic bee paralysis is a little bit different. It's not very common. The AKI viruses are much more common, but you still see those shaking and shivering bees. Crawling bees on the ground gets described a lot. Those dead bees below the entrance. Unique to CBPV is this description of hairless black shiny bees. One thing I like to note with this is that especially if you use mineral oil beetle traps, to be sure you know what you're looking at if you see what we would call these shiny dark bees, because on a couple of occasions, beekeepers who thought that this is what they were seeing had actually spilled oil from their beetle traps, and it can look very, very similar, which is super interesting. This one is not super strongly associated with Varroa, but high infection levels can absolutely cause a colony loss. And as I mentioned, it can look a lot like a pesticide kill, depending on how things go. Finally, we have the Lake Sinai viruses. These are a relatively recent discovery. There's quite a few strains, at least 7 that I'm aware of. And we don't understand them that well yet. We discovered them by a genetic test, and so we know what their genetic code is, but we don't know that much about what they really mean for the bees. We do know that they're very, very commonly detected, and that the levels are typically higher in weak or dead colonies. But there's a lot more work to be done on these Lake Sinai viruses before we really know more about them. So it was mentioned at the beginning, I work with our Queen and Disease Clinic at the lab. This is our extension effort to not only educate about these viruses, but also to do testing for these different diseases. So we do detection of honeybee viruses and other pathogens. This is a service offered to beekeepers at all scales for a certain cost. And when those samples come into us and we do a test, we compare those samples to our research database. We have a pathogen panel that we always run. It includes that acute bee paralysis virus, black queen cell, chronic bee paralysis virus, 2 strains of deformed wing, Israeli acute paralysis virus, Lake Sinai virus, and then 2 non-viral pathogens. It's going to be a trypanosome panel that's going to pick up Cryphidia, which is a gut parasite, and then a nosema that's going to pick up Nosema apis or Serrana. Sac brood, which I mentioned to you guys, and some of the other brood diseases, including foul brood, are not currently included in our panel because we sample adult workers, and the best way to sample for brood diseases is to sample brood. But because viruses especially break down really readily at regular temperatures or even in regular freezers, we haven't yet found a way to effectively get good samples of brood from beekeepers to us in a way that isn't cost prohibitive for the beekeeper. So that's something we're really interested in expanding into, but we don't currently offer it. So this is our research database as it currently stands. We have over 1,300 colonies represented here. I'm not going to talk to you guys about those 2 non-viral pathogens today because we're pretty focused on viruses for this. What you're going to see on this vertical axis is the percent of colonies tested. So wherever this bar falls, it's based on percent of total colonies. For example, approximately 40% of the colonies we've tested have been positive for deformed wing virus A. The colors there then indicate high, medium, or low levels of infection. So that darkest red is the highest level of infection. So again, for example, 10% of the colonies we've tested have had high levels of deformed wing virus A relative to the other samples in this database. As you can see here, deformed wing is one of the most common pathogens we detect, which makes sense. That's why it gets talked about so much. That's why it's such a problem for beekeepers. It's pretty common. And importantly, as we receive new samples, our database continues to improve. You know, these viral trends can change over time and things like that. And so the more samples that we get over time in a specific time point, all of that helps improve our database, and it helps us give beekeepers better information about where they fall sort of on a sliding scale of viral infection. The kinds of questions that we ask about viruses as researchers, a lot of these are very focused to projects that we are doing or have done recently in the lab at NC State. First of all, what viruses are detected the most often in colonies and at what levels are they present? I just talked about that a little bit with our clinic database, but we also apply that to other work that we do. What kinds of viruses do we detect in queens specifically? Are they infected by the queen mother or the workers that tend them? What kinds of viruses do we detect in drones, and how does this affect their fitness and ability to mate? How do other stressors impact viral levels and vice versa? Dr. Joe Malone, who just graduated and has moved on to the EPA, looked at pesticides specifically and how they interacted with viruses. Which of these viruses are found in native pollinators? Do shared floral resources cause virus transmission? Hannah Levinson, who is still with the program, she's a PhD student, is looking at that specifically with native pollinators. And then finally, what viruses do we find in mites and how does that affect the colony? That's what we're going to talk about with the research project today. So how do we typically study viruses in mites? And this "we" here refers to the research community again. Virologists especially tend to look at these viruses as being very disparate from the bees. They'll study a single virus in individual mites or individual pupae and their associated 5 or 6 mites. They're interested in the way the viruses are transmitted, which, as I mentioned, is very important. They're interested in whether these viruses replicate actively in the mites. They very rarely look at this at the colony scale. It can be pretty difficult. There's a lot of noise there and things like that, which you'll see as we get to the data. And typically, researchers look at focused questions and not doing holistic analysis, which is very important because we really can't know that much about these viruses without these focused questions. But at some point, we then have to expand that to what we would call a field realistic situation. And so that's how we started developing this research paradigm. And so what I have here is just a chart of interactions. We know that mites parasitize bees in the colony. We're going to give this interaction a term. We're going to call it MC, mites in the colony. We know that viruses infect and or are vectored by mites. So virus in the mites is VM. We know that viruses infect bees in the colony. So virus in the colony is VC. We monitor these terms to inform management already as beekeepers. A sugar shake, for example, is used to measure mites in the colony, or an alcohol wash, or a sticky board. So that's a measurement of MC. We already know that in many cases, as mite levels increase, levels of virus in the colony also increase. So we see a positive association between MC and VC. To manage this, we control mite levels in order to control virus levels in the colony, with the hope that lowering MC will also lower VC. Similarly, beekeepers can send samples to our Queen of Disease Clinic for reports on their colony viral levels. It's a direct measurement of colony health to measure this term VC. In some cases, colonies will have high viral levels even when mite levels are below treatment threshold, which led us to the question, what's the viral level in the mites? This term VM is getting left out of those measurements. This project aimed to look at all 3 of these terms simultaneously. We're interested in the relationships between mite levels and viral levels, and previous studies have really rarely looked at all of these interactions simultaneously, not only in a single bee, but in a colony. And so our goal is to fill in this interaction map. So what you're seeing here is a representation of these relationships and their significance. Which of these lines should be the strongest between these different terms? And so we developed an experiment. I absolutely cannot talk about this experiment without talking about Ashley Rua. She was an undergraduate researcher with us at the time. She has since graduated and moved on to the vet school at NC State. But she was a huge part of the work for this project, and I absolutely could not have done it without her. So what we were interested in with this experiment was, what viruses do we detect in colony samples and Varroa samples? How do Varroa counts, Varroa viral levels, and colony viral levels associate? And ultimately, what drives colony viral levels? So what we did is we collected samples at our NC State apiary and then also from hives from a local beekeeper. We performed a total of 4 rounds of sampling at different times of the year. One of the things that we looked at was bees that were taken directly from our sugar shake versus bees that were taken from a separate brood frame in the colony, and I'll talk a little bit more about that. We used sugar shakes to collect all of our mites. And as is standard, we would take a half cup of bees, which was about 300, and we would put them in our sugar shake jars, and then we would wait several minutes and then invert and shake for mites. And we would collect those specific mites from that sugar shake sample. And then we would additionally collect 50 bees for viral analysis. We tested our round 1 mites separately, as in each individual mite got tested for its viral levels. But in the rounds following that, we pooled them for expediency and cost reasons. And we also pooled all of our bee samples, which is standard practice with our viral measurements in the lab. We extracted a genetic material that's called RNA. All of these viruses that we're interested in are RNA viruses. We then synthesized the complementary DNA because in order to do the kind of genetic testing we need to do, we can't work with the RNA directly. And then we used a test called qPCR, which lets us measure the absolute levels of virus of interest in those samples that we started with. Ultimately, we detected deformed wing A, deformed wing B, and IAPV in both the bees and the mites for all 4 rounds. There were other detections in just bees or just mites, but we focused on the viruses that they had in common. Unfortunately, our IAPV detection was too infrequent for analysis, so I won't talk about it much. Our mite counts varied anywhere from 0 to 75 per 300 bees, which means our mite level percents were anywhere between 0 and 25%. Typically, treatment threshold is described as somewhere between 2 and 4%. So I mentioned these sugar shake bees versus brood frame bees. So most of our samples are, we took the sugar shake and the bees are from that peanut butter jar and the mites are also from that peanut butter jar. But something we were interested in was if we took the mites from the peanut butter jar but took the bees from a different frame, would the relationship still hold true, or was it necessary to get the mites from the specific bees you were testing? And so what you're looking at here with this graph, I don't know if I have an annotation. So on one vertical axis, we have the bees that we took from the peanut butter jar. On the bottom horizontal axis, we have the bees we took from the brood frame. And what this chart is representing is the amount of deformed wing virus A those bees had. Ultimately, what we're looking at here with this positive association is that it held true no matter where the bees came from. If the bees in the peanut butter jar had a lot of deformed wing, the bees from the adjacent frame did as well, and vice versa. If the peanut butter jar was low, the brood frame bees were low. So that was really encouraging to us. It told us that this disparate sampling was still relevant, and it also told us that these viruses were really systemic in the colony, that the trends that we see on one frame, we're also going to see 3 frames over. Ultimately, this is a measure of sampling effectiveness. You don't have to sample the actively parasitized bees to accurately measure the viral levels. So these individual mites from round 1 were super interesting. Everything on this chart on the left, this blue chart, those mites came from a single sugar shake. They only represent 1 colony. And so what you're looking at are 20 different mites, which came from 300 bees. Each cell here represents 1 mite. And on the first row, you're going to see deformed wing A. The second row is deformed wing B. And just like in that earlier chart, a lighter color is a lower infection level, a darker color is a higher infection level. They're highly variable. You know, we didn't see that all of the mites from the same sugar shake had similar viral loads. Some of these were 0. So if, for example, I had only looked at this mite for this colony and hoped that it would tell me the story of what was going on there, I would have been severely misled. I would have thought that there was no deformed wing present when in fact that wasn't the case at all. Similarly, this mite here had super high levels of deformed wing A and no deformed wing B. That mite alone also could not have told me the whole story. So what this told us is that since these are so variable, the best way to get a snapshot of what's going on in the colony is to pool those samples. So instead of extracting every single mite for every single sugar shake we did, we collected them all into a single sample, and what you ultimately get is an average. So here are our viral results. This chart is a little bit intense, and I'm going to walk you through it. What you're seeing here are 3 different representations of our results, our sampling results. This axis here shows values 0 to 4. 0 is no virus or mites, depending on which chart you're looking at. 1 is low, 2 is medium, 3 is high, and 4 is very high. A similar scale to what we've been working with up to now. The first graph is the viral level in the bees. Each of these individual points, you see these numbers along the bottom, is an individual colony. So we had 90 colonies total. The second graph is then the viral level in the mites, and these graphs all line up. So for example, if you're looking here and you follow along this axis, this is colony 50, that's on a line, that's easy to see. So this is going to tell you the viral level in the bees in colony 50. This is the viral level in the mites in colony 50. And then finally, we have the mite level in the colony, which was taken as the number of mites in that sugar shake versus 300 bees. So what we are looking for, if wisdom held true, is that all these graphs would look very similar. Mite levels and viral levels would rise and fall together. But what we see instead is a lot of variation, which is why at first glance this is so noisy and it's so difficult to pull patterns out of. So here's an example where we have a high mite count with low mite virus and low bee virus. So this green graph here is relatively high, but we've got zeros for the amount of virus. So in spite of that high mite load, we're not seeing a viral stress. Alternatively, here we have a low mite count, not a zero, but a low, but we've got moderately high and very high levels of virus in the mites and the bees respectively. So again, that doesn't hold with the relationship that we would expect, high virus with high mites. Here's a very specific example where treatment threshold is typically about this 2 level, this medium level. So these mites are below treatment threshold, but we're seeing a significant viral stress on that colony and very high viral levels in those mites, in spite of the fact that there weren't that many of them. So what this tells us is that a lot of the times we get a question, I treated for mites, my mite levels were low, why did I still lose the colony? This is one potential explanation, is that you have a viral stress that is at odds with what you would expect with your mite load. So this all leads back to this paradigm that I presented in the beginning. All of these relationships, which is the most important? What's the driver of colony health? And so here's another way to look at the data. This is just deformed wing A. Everything I presented to you at this point is just deformed wing A. So this first graph is viral load in bees and mites. This tells us that as the amount of virus in the mites goes up, the amount of virus in the bees also goes up, also vice versa. We have another positive relationship. Next to that, we have the viral loads in the bees on the horizontal axis, and then the mite counts. And you notice that that positive relationship is not as strong. That viral relationship has a stronger positive line than the mite count viral relationship. We saw some very similar results for deformed wing B. I don't like to talk about just one set of results. I really like to present all the data so everybody can see that these trends sort of held true. This is those same 3 graphs just overlaid with each other. Again, if we saw what we expected to see, all of these lines would rise and fall together. They would almost become indistinguishable because they would all fall in line. And that's not what we see. We see a lot of noise, which is the takeaway. What you're seeing here is a lot of noise. This green line, again, is the mite loads, and it's all over the place compared to those viral loads. And again, we see that same relationship where the positive correlation of viral loads in bees and mites is stronger than the positive relationship in bees and mite counts. So that led us to fill in this graph that I talked about at the beginning. So what you're seeing here, first of all, is some dashed lines. Those indicate no significance. So I told you at the beginning, IAPV was not detected often enough to analyze effectively. That's why we don't see any significance in those blue lines. IAPV was kaput. The thickness of the line indicates the strength of significance, the strength of that relationship. So thicker lines are stronger relationships. For both strains of deformed wing, A is black and B is red, we can now see that the mite levels in the colony, MC, do not drive the viral levels in the colony. For A, there's no significance, and for B, there's the least amount of significance out of all these relationships. So MC to VC does not seem to be the driver that we would expect it to be. However, viral levels in the mites have a strong positive relationship with viral levels in the bees. You can see that A line is very dark black with very thick black line, which makes sense given those charts I showed you a minute ago, those scatter plots. It's also interesting to note that as this MC term, mites in the colony, increased, virus in the mites also increased. This isn't necessarily a result we expected, but it also makes sense when you start to think about it. One possibility is that this increase in viral levels is a result of a weakening colony. So as the mite levels increase, the colony tends to get weaker and tends to have more of a viral stress. It's also possible that as the bees get sicker, the mites feed on more sick bees, either more number of bees that are sick or more individual bees with higher sickness in themselves. We don't know yet. That's not something that shakes out from this particular experiment, but it was an interesting result to see that positive relationship there. And so our conclusion really comes down to a lot of what we already knew, which is that mites don't kill colonies. Viruses kill colonies. And since that mite viral level is a stronger driver for colony viral level than the mite counts are, the mite count can't tell us the full story. they're still important, especially as a backyard beekeeper. It's one of the only tools that you have to estimate what's going on in your colony in terms of viral stress. When viral levels in bees are what we're interested in, though, and colony health is what we're interested in, measuring mite levels is a proxy of a proxy, because what you really want to know is virus in the colony. That's the end game. So mite counts lead to virus in the mites, lead to virus in the colony. It's a proxy of a proxy. Viral measurement can be really important for identifying colony challenges. I touched on that when I talked about the paralysis viruses, where you might assume that you have a pesticide kill, but you actually have a viral stress that's going on. And so we're interested as researchers and as beekeepers, what kind of implications this data can have for management and for colony health. But with any study comes limitations. Right out of the gate, IAPV was detected too rarely to draw any conclusions, and other than that, we only had deformed wing to fall back on. We didn't find any other viruses in the mites, and so we can't extend these conclusions to any virus other than deformed wing. We only sampled colonies in Wake and Moore County and only in North Carolina, so it would be interesting to see if these trends hold true outside of our immediate area and across the country, especially in areas with different climates. This does not account for replication of virus in mites, which I touched on earlier as something of interest to virologists. That's not something we tested for at all. It does not take honeybee stock into consideration in any way to see if these relationships change if you're not dealing with Italians, for example. And really importantly, the expense of virus testing creates a barrier for beekeepers. So it's easy for us to say everyone should test their viral loads, but it's not easy, it's not fast, and it's not cheap, which is a problem, especially as you're talking about a whole lot of colonies in your operation. Some directions that we're interested in taking this, at what level does deformed wing virus become problematic for colonies, and when is it lethal? And interestingly, we want to know if we can answer those questions for the viral titers in the mites. So can we take that proxy of virus in mites and use it to extrapolate when a colony is in trouble? We're interested in how different forms of mite control affect mite viral levels. So we know how different forms of mite control affect mite counts, but how does it affect these viral levels? We want to know if colonies can have high mite levels and low viral levels, and we want to know if that might be an interesting stock to breed. You know, if they're able to maintain these high mite levels without a viral stress, is there a genetic component to that, and can we capture it? And interesting is what other stressors selectively bred bees might be susceptible to. So if we do go down that avenue, what new weaknesses do we open up because we're selecting for something specific genetically and we don't know sort of what cart comes with that horse? Again, I want to mention Ashley Rua, who was a huge part of this project. Also, Jen Keller's contribution cannot be understated. I absolutely could not do any of this work without her. The rest of the lab also helped humongously, especially when it comes to sampling. Both Joe and Lauren, who have both now left the program, were huge parts of this project, and our funding was through USDA NIFA. And with that, I will be able to take some questions. You guys are welcome to unmute or to use the chat, whatever you're comfortable with.
Hi, Erin. This is Lori.
Hi.
Hey, I really enjoyed this. It is fascinating, everything that you all are learning about the viruses.
Thank you.
Yeah, I know it opens up a lot of questions too. And my question is, if it's not necessarily the mite counts, but the viral counts, how do we affect that?
Yeah, that's a really good question. And it's a question we want to pursue. The short-term answer for you today is that the best thing you can do as a beekeeper is still to control Varroa. If you have low Varroa counts, that is still going to be strongly associated with lower viral stress. We're hoping at the very least that a lot of those other cases are outliers. Ultimately, we hope to move towards a place where we might be able to affect those viruses more directly, whether it's through genetic modification or supplements or something of that nature. We're just not there yet. So we're learning more about what we need to measure, and then what comes after that is how we're able to affect what we measured.
Okay, thank you.
Yeah. So I have a question in the chat here. I'm going to read it out in case anybody can't see it. It says, it looks like you use the sugar shake method of sampling. Have you found that to be the same accuracy as alcohol wash? So the reason that we use sugar shake specifically is that different kinds of alcohol can be a problem for the genetic testing we use downstream. And so we didn't want to introduce alcohol into our samples on the off chance that it would affect our results. Sugar is pretty inert for our purposes here. That said, 2 points for the difference between sugar shakes and alcohol wash. Done correctly, a sugar shake is accurate enough for any sort of management purpose. That said, that requires that you do let those bees sit in that powdered sugar for a given amount of time to give those mites time to fall off, that you do shake pretty vigorously to be sure all the mites are dislodged. Those are some things you don't have to worry about with an alcohol wash, which in that way makes it a little bit more user-friendly. Evidence up to this point does indicate that alcohol wash is the most accurate. But at the end of the day, the most important thing to consider is what you will realistically use as a beekeeper. A sugar shake done correctly is infinitely better than no testing at all. So if you don't have alcohol on hand, if you're not comfortable with it, if you don't have the apparatus that you need, use the sugar shake because it's going to give you more of a picture than you would have had if you didn't count at all. But do make an effort to do the sugar shake correctly because it's going to give you a much better sense of what's going on than if you rush it or you skip steps or anything like that. That's a really good question, and it's one that I get a lot about the difference between sugar shakes and alcohol wash. Even in my own hives, I typically do sugar shake just because it's so easy. I keep powdered sugar in the back of my car at all times, and anytime I want to stop by the bee yard, I'm ready to go. So I think it's a really effective tool.
Hey, I just wanted to comment. Your research bums me out.
Oh, I do get that a lot, especially when we had a bigger lab. A lot of our other talks were a lot less of a downer. As the virus person, I tend to be a downer. The good news is that the more we understand, the more we're going to be able to do to make it better. So I understand though, it is a bummer. I can appreciate that.
Erin, I've got another question. The Asian bees, as far as they're being able to tolerate the mites, is there any research to suggest that they're tolerating the mites or they're adapted to the viruses?
That is a really good question that unfortunately off the top of my head, I don't know the answer to. I would have to look into that. I do know that the Varroa stress is not as significant on that sort of in that native relationship between Asian honeybees and Varroa mites. But as far as the viruses that are associated with it and how that relates, I don't know. If you want to shoot me an email, my email address is on our website. Our website is ncsuapiculture.net. And under the people page, I'm listed under my name, Erin McDermott. Shoot me an email and remind me of that question, and I would be more than happy to look into it for you and get you an answer because I'm also interested in the answer to that. I don't know off the top of my head.
Okay, thank you.
Yeah, absolutely. I've got another question in the chat. It says, how much is the testing you referenced that determines viral load? Our current cost is $55 a sample, and a sample in this case is 1 colony, typically. That's a pooled sample of 50 bees from a single colony. And then in addition to that, there's the cost of getting the bees to me, which have to be shipped live, which involves a right-way shipper, and the cost of overnight shipping. So $55 is what we charge, and then additionally you have to consider getting bees to me. And we do have a 5 colony minimum on top of that. That said, especially as a club, if you guys were ever interested in doing viral testing and wanted to pool samples, say for example, you only have 2 colonies, but your neighbor has 3, you would be able to pool those to meet that 5 minimum and things like that. But as I know it's not cheap and it adds up pretty quick, especially with that 5 colony minimum. Pat sent a question that she wants answered. I don't see a question from Pat. So Pat, if you could send that to me again, I'm not seeing it in the chat. So just type it for me one more time. I have another question. Is there any chance of a beekeeper administered viral load test being developed in the future? I would say absolutely. I've seen a little bit of work done on what we call ELISA tests, which if anybody's familiar with the foul brood test that you can buy, that you can sort of use right at the site of your colony to test for foul brood, I think that's what a lot of people think of when you're talking about this disease testing. There's absolutely interest in developing things like that, especially for deformed wing virus. Foul brood is a fungal or bacterial or whatever infection. It's a little easier to develop those tests. Viruses are just genetically different. It involves like a development of antibodies and things like that. But the interest is absolutely there. And I'm certain that there are a number of companies in addition to academic institutions that are actively working on it. So I would not be surprised at all if within the next 5 to 10 years, you started to see products like that emerge. This question says, have seen higher mite counts this year, but not seen signs of viruses. In the past 4 years at 4Cs, the viruses were very visible, yet mite counts were low. What are you seeing? I am also seeing higher mite counts this year. At this point, I'm talking about the colonies that I work with personally. Jen Keller would be better suited to answer questions about our research colonies. As you can imagine with COVID, I didn't get to do much with research colonies this year. As far as signs of virus, I gotta be honest with you, I don't typically see a lot of visible signs of virus. Part of that is because I'm a very aggressive mite controller. As you might imagine, doing this research for any length of time, it makes you very paranoid about your mites. So personally, I haven't seen any signs of deformed wing or anything like that, but I also typically don't. That would be a really interesting question for the inspectors, especially the inspectors in your area specifically. You could even shoot an email and ask just sort of what their thinking is on that, because they go through colonies, you know, all day, every day, and they would know the answer to that. That's an interesting question. I'm going to have to keep that in mind for our next apiculture online webinar, see if I have time to ask the inspectors that, because I like that question a lot. My next question says, do you know of any research being done on treating colonies with antiviral medications? I don't personally remember reading any papers, but I would be shocked if no one was looking into it. The thing about the way research publication happens is, especially something like that, which is a large scale project, it takes time. And unless you happen to run into that researcher at a conference, and right now conferences are not happening, you might not even know that that research paradigm is of interest to them. But then when the paper comes out, like 2 years after the experiment's finished and they've had a chance to write it up and submit it and have it accepted, that's when everybody gets to get excited about it, right? Because they have results. So I don't know of anybody who's working on it, but I am sure that somebody is. The difficulty there is that there are so many different kinds of viruses in honeybees. So you might be able to administer an antiviral that worked on, say, deformed wing, but it wouldn't cover something else. So you have to be sure you're not opening up a new susceptibility, like you're creating a vacuum or something like that. Like there's a lot of elements at play that you have to make sure you understand before you make any kind of recommendation to beekeepers, because you don't want to make a bad thing worse. Did I get to Pat's question? Good. I was hoping that it would come through. These are really excellent questions. I really appreciate that, you guys. This is my favorite part. I love answering questions. Anybody else?
I'm going to ask one by voice here.
Yeah.
So you had 50 different colonies that you were testing. You said they were across 2 different counties, Moore and Wake. I'm assuming they were from different sites within those counties. Was there any correlation, like from site to site, saying that this site had more deformed wing A?
Gotcha. So we don't have the answer to that question. So I mentioned 4 rounds. This is the difference between what the paper says and what really happens. It makes it sound like we planned that from the beginning and we were always like, we're going to have 4 rounds of sampling. That's not how this went at all. We started with our first round, realized that it was interesting, and then we were like, we should do this again. So then in a different season and at a different place, we did a 2nd round. And then we were like, oh, well, we didn't try this thing. So then we had a 3rd round and a 4th round. And so the reason they're not comparable is because they were at different times. So you can change your space or your time and make comparisons. But if you change both, for example, you're testing different colonies in the spring than you are in the fall, you can create trends overall because now these are all different colonies, but you can't make comparisons between them because you don't know at that point if the difference is time or space because you changed both. Does that make sense? So that would be something that would be interesting to look at. We would just have to design a different experiment to answer that question. This one doesn't have the kind of data we need to answer it. We already know we see changes over season, which is part of the reason we had to pool all 90 of these colonies, because if it was any fewer than that, the noise would be too much to draw any conclusions. So the fact that this holds true in spite of all those differences is interesting in and of itself. But that's part of why this kind of research, as opposed to what I described earlier as pure virological research, is messy. And you have to be careful not to draw conclusions that don't exist because it's messy. Anybody else? Did I miss anything in the chat? I don't think so. Awesome. Well, thanks you guys so much. I really appreciate your great questions. I appreciate your attention. I know especially via Zoom, it can be a little tough. So I really, really appreciate it. If anybody does think of any more questions, if you want to reach out about the clinic, if you want to reach out about our research program, find us on Facebook or on our website, which is ncsuapiculture.net. And be sure to check out our webinars, which have gotten pretty popular, and we're having some cool guests coming up. So we have our apiculture online webinars every other Wednesday. Typically, we did skip the previous one because Dr. Tarpey was indisposed. He was meeting with big government bigwigs. So this coming Wednesday, a week from yesterday, we'll have our next one, and we post the link to our YouTube and to our Facebook channel. So definitely check us out there. Thanks so much, everybody, for your lovely messages. That's so nice. Thank you very much.
Thank you, Erin.
Thanks, you guys. I think I'm safe to stop the recording. I'm going to stop it now.